Hello everyone, my name is Rahul. In the previous video, we had discussed about endomembrane system. In this video, we are going to start with the first component of endomembrane system, that is endoplasmic reticulum. Endoplasmic reticulum, it is a network of membrane-bound tubules inside the cytoplasm. The word endo means inside, plasmic means cytoplasm, reticulum means network. So it is a network of membrane-bound tubules which is found inside the cytoplasm. Now there are three components of endoplasmic reticulum. First is cisternae. The second component is vesicles. And the third is tubules. So the endoplasmic reticulum, it can be arranged in the form of cisternae, that is flattened vesicles, or it can be present as vesicles, or it can be present as cylindrical structures which are called as tubules. So cisternae are long and flattened. They are long and flattened, unbranched units arranged in stacks. Vesicles are oval membrane-bound structures and tubules, these are irregular branched cylindrical structures and they are also membrane bound. The difference between endoplasmic reticulum and Golgi body is that these components are present isolated in the cytoplasm and are not arranged in form of a complex. Now, the presence of ER divides the space inside the cytoplasm into two compartments. One is the extraluminal compartment, that is between membrane of endoplasmic reticulum and plasma membrane, and the second is intraluminal, that is space present inside the ER. So ER is basically a network of spaces bound by membranes. So the space present inside the ER is known as the intraluminal compartment. Now there are two types of ER depending upon whether ribosomes are present or not. One is rough endoplasmic reticulum, the other is smooth endoplasmic reticulum. So ribosomes, which are of 80S type, are bound by their larger subunit to the membrane of endoplasmic reticulum. So when ribosomes are present, it is known as rough endoplasmic reticulum. In smooth endoplasmic reticulum, ribosomes are not seen. These ribosomes, they are attached by the help of two proteins known as ribofourin 1 and 2 on the surface of rough endoplasmic reticulum. Now, The rough endoplasmic reticulum, it is mostly made up of cisternae, and in smooth endoplasmic reticulum, it is mostly made up of tubules. So cisternae are more in rough endoplasmic reticulum and tubules are seen more in smooth endoplasmic reticulum. In rough endoplasmic reticulum, it is abundant in cells which are actively engaged. It is abundant. It is abundant in cells which are actively engaged in protein synthesis and secretion. The smooth endoplasmic reticulum, it is abundant in cells concerned with glycogen and lipid metabolism. So it is abundant in cells concerned with glycogen and lipid metabolism. So in animal cells, most of the lipids like steroidal hormones, they are synthesized by the smooth endoplasmic reticulum. So example, adipose tissue and adrenal cortex, interstitial cells, etc. Examples in rough endoplasmic reticulum where they are abundantly seen are liver, pancreas, goblet cells, etc. This is how a endoplasmic reticulum appears. The rough endoplasmic reticulum, as you can see, it is continuous with the membrane of nucleus. So rough endoplasmic reticulum, it is continuous with the outer nuclear membrane, whereas smooth endoplasmic reticulum, it is not continuous with the outer nuclear membrane. And you can see the presence of ribosomes on rough endoplasmic reticulum, whereas in smooth endoplasmic reticulum, ribosomes are absent. Now, there are two modifications of ERs seen in cells. One is known as sarcoplasmic reticulum. Sarcoplasmic reticulum. This is seen in muscle cells or muscle fibers and also in cardiac muscles. Sarcoplasmic reticulum acts as store of calcium ions and energy rich compounds needed for muscle contraction. The second modification is known as microsomes. They are pieces of ER associated with ribosomal particles. These can be obtained by fragmentation and high-speed centrifugation of cell. They microsomes do not exist. These microsomes, they do not exist as such in the living cell. Now we move on to the functions of endoplasmic reticulum. First, it acts as the endoskeleton of cell, thereby providing mechanical support to the cell. The second function is intracellular transport of substances. Since it is forming an extensive network inside the cell, so if any substance is to be moved inside the cell from one place to another, it can be moved or transported with the help of endoplasmic reticulum. Third function is rough endoplasmic reticulum acts as factory of protein, that is protein synthesis. Protein synthesis, it is done by rough endoplasmic reticulum due to the presence of ribosomes on its surface. Smooth endoplasmic reticulum is involved in synthesis of lipids and steroidal hormones. Smooth endoplasmic reticulum is also involved in detoxification of various exogenous and endogenous substances. For example, alcohol, drugs, etc. They are detoxified with the help of smooth endoplasmic reticulum. The next function is endoplasmic reticulum provides an increased surface area for cellular metabolic reactions. It provides an increased surface area for cellular metabolic reactions. Next function is, along with Golgi apparatus, endoplasmic reticulum is involved in biosynthesis and repair of cell membrane. So it is involved in the biosynthesis and repair of cell membrane. It is also involved in synthesis of lysosomes, Golgi, and some microbodies. Now, and the next function is that endoplasmic reticulum, it is providing, ER provides the precursor of secretory material to Golgi bodies. So whatever is secreted by the Golgi bodies is actually produced by endoplasmic reticulum. So with this we finish off with our discussion on endoplasmic reticulum. See you in next video.